You wake up, your mouth tastes like something died. That smell, that taste, you know it. Everyone knows it. Morning breath, the thing nobody talks about at breakfast. The reason you brush your teeth before you kiss anyone. But here's what they don't tell you. What's actually happening in your mouth while you sleep is stranger and more disturbing than you'd ever imagine. Your mouth isn't just resting. It's hosting a bacterial feeding frenzy that would make a horror movie director proud. And the chemistry behind that smell, the actual molecules responsible, they're the same compounds used to detect gas leaks, and they're toxic enough to damage your cells at high concentrations. Let me walk you through what science has actually discovered about this. No myths? Just the documented facts from labs that study oral bacteria by the thousands and chemists who measure these compounds down to parts per million. Because understanding what happens in your mouth at night, that's where it gets really weird. Your mouth right now contains somewhere between 500 bacterial species and 700 depending on whose research you read. The Human Oral Microbiome Database, launched by the National Institute of Dental and Craniofacial Research, documents over 700 species of prokaryotes belonging to 185 genera and 12 major phyla. That's more bacterial diversity than almost anywhere else on your body except your gut. A 2005 study published in the Journal of Clinical Microbiology, analyzing samples from 5 clinically healthy subjects across 9 different oral sites, found 141 predominant species just in those individuals. Over 60% had not been cultivated in laboratories. They're known only as phylotypes, genetic sequences without names. Bacteria, we can detect but not yet grow. Each person harbors somewhere between 34 and 72 different varieties at any given time, according to research tracking multiple subjects. Your teeth alone carry between 1,000 and 100,000 bacteria per tooth, depending on how often you brush and floss. When you multiply that across 32 adult teeth, the numbers become staggering. Millions! Tens of millions, possibly hundreds of millions of bacterial cells living in your mouth right now as you read this. During the day, these bacteria are manageable. Your saliva is flowing at 0.3 to 0.4 milliliters per minute when you're awake and not eating, according to a 2025 meta-analysis published in the Journal of Dental Research that reviewed 3,167 healthy adults. That's about half a liter total per day, 500 milliliters. Your saliva isn't just spit, it's a biological weapon system. It contains antimicrobial proteins. It washes away food particles. It dilutes bacterial waste products. It keeps oxygen levels high enough to suppress certain types of bacteria, the anaerobic ones, the ones that thrive without oxygen. The dissolved oxygen in saliva directly kills certain bacterial species that cannot tolerate aerobic conditions. This constant flow creates mechanical shearing forces that prevent bacteria from establishing permanent colonies in some areas. The proteins in saliva, including immunoglobulin A and lysozyme, attack bacterial cell walls. Lactoferrin in saliva binds iron, making it unavailable to bacteria that need it to grow. But when you fall asleep, something changes. Your body signals your salivary glands to slow production dramatically. Research from 1956 by Schneier and colleagues, published in the Journal of Dental Research, found that during sleep the salivary flow rate drops to almost nothing, virtually zero. Some papers say negligible. The exact mechanism makes sense from a survival standpoint. If your mouth produced saliva at normal rates during 8 hours of sleep, you'd either be swallowing constantly, which would interrupt sleep, or you'd lose massive amounts of water through drooling. Evolution solved this by shutting down the taps. But evolution didn't account for the bacterial consequences. When saliva production drops, your mouth dries out. The medical term is xerostomia when it's chronic, but everyone experiences a mild version during sleep, that dry mouth. Without the constant wash of fresh saliva, several things happen simultaneously. Food particles that were stuck between your teeth or in the grooves of your tongue stay there. Dead epithelial cells that your mouth sheds naturally. They accumulate. White blood cells that died fighting bacteria in your gum tissue, they pile up. And the bacteria that were being held in check by flowing saliva and high oxygen levels, they multiply fast. The tongue is the main source. Research published in Anaerobe in 2003 by Tyrrell and colleagues, who cultured tongue samples from 8 people with documented oral malodor, found specific species in 62.5% of subjects. Peptostreptococcus anaerobius, Fusobacterium nucleatum. Pigmented Prevotella species, Veillonella species in 100% of subjects. These aren't exotic bacteria. They're normal residents of your mouth. But at night, when conditions change, they go into overdrive. Studies measuring bacterial counts in saliva using colony-forming unit methodology have found concentrations ranging from 3.7 times 10 to the 5th power CFU per milliliter for Streptococcus mutans in healthy adults. That's 370,000 colony-forming units in each milliliter. For lactobacilli, another common oral species, the average was 3.8 times 10 to the 5th power CFU per milliliter, 380,000 per milliliter. And that's just 2 species out of 700. Research examining bacterial counts over time shows that numbers increase markedly during sleep. Though exact multiplication rates vary by species and individual conditions, the bacterial load in morning saliva is consistently higher than evening saliva. The bacteria don't just increase in number, the composition shifts. Certain species that were minor players at night become dominant by morning. The 12 bacterial phyla that colonize your mouth represent an ancient evolutionary partnership. Firmicutes, fusobacteria, proteobacteria, actinobacteria, bacteroidates, chlamydia, chloroflexi, spirochetes, SR1, synergistetes, Saccharobacteria, which used to be called TM7, Gracilobacteria, which used to be called GN02. Of the approximately 700 species identified, about 54% are officially named. 14% are unnamed but can be cultivated in laboratories. 32% are known only as uncultivated phylotypes. DNA sequences without living cultures. The species common to all mouth sites belong to genera, including Gemella, Granulocytella, Streptococcus, and Veillonella. These core species establish themselves early and persist, but different surfaces favor different communities. Your tongue, with its papillae and fissures, harbors more diverse and more anaerobic species than your smooth buccal mucosa. The pockets between your teeth and gums, when healthy, contain different species than the visible tooth surfaces. Each ecological niche in your mouth supports specialized bacteria adapted to that exact environment. If you value documented microbiology and biochemistry, like this subscribe, it's the best way to support our work. Also, comment from which city you're hearing this from. This script is researched and factual using sources from peer-reviewed publications in journals like the Journal of Agricultural and Food Chemistry, published by the American Chemical Society in 2017, studying volatile sulfur compound chemistry by Kreitman and colleagues, and PLOS Wund. Published in December 2021 by Shimizu and team examining how sleep affects oral biofilm microbiomes with 16S ribosomal RNA gene sequencing on 10 healthy subjects over 7 days with samples from 6 oral sites each. Now back to the information. The dorsal surface of your tongue has a unique structure, filiform papillae, little finger-like projections, fissures, crypts, deep grooves. A 2024 study in Scientific Reports by Park and colleagues from Korea using bacterial biofluorescence imaging technology called quantitative light-induced fluorescence showed that these deep fissures create low oxygen conditions, anaerobic environments, perfect for the gram-negative anaerobic bacteria that produce the worst smells. The paper documented that 85% of halitosis cases are caused by volatile sulfur compounds produced specifically by these anaerobic species, Porphyromonas gingivalis, Treponema denticola, Veillonella species, Fusobacterium nucleatum. These bacteria don't just sit there, they eat their food, proteins, specifically sulfur-containing amino acids. 2 of them matter most, cysteine and methionine. These amino acids are everywhere in your mouth. In the dead epithelial cells sloughing off your tongue and cheeks, in the remnants of your last meal, in your saliva itself, which contains multiple proteins, in the white blood cells that fought and died in your gum tissue, in blood components that seep from your gingival crevices. When anaerobic bacteria break down cysteine and methionine, they use specific enzymes. Cysteine desulfidrase cleaves cysteine releasing hydrogen, sulfide. Methionine gamma lyase breaks down methionine producing methyl mercaptan. These aren't simple chemical reactions. They're sophisticated enzymatic pathways that bacteria evolved over millions of years. The bacteria transport the amino acids into their cells through specific membrane proteins. Inside the cell, enzymes bind to the amino acids. The enzymes break carbon-sulfur bonds. The sulfur-containing fragments are released as waste. The bacteria expel these volatile compounds because they're toxic, even to the bacteria themselves at high concentrations. The result, clouds of sulfur gas permeating your mouth, volatile sulfur compounds. VSCs in the scientific literature. 3 compounds dominate: hydrogen sulfide, chemical formula H2S, methyl mercaptan, chemical formula CH3SH, dimethyl sulfide, chemical formula open parenthesis CH3 close parenthesis 2S. These three molecules are what you smell when you wake up. Let me be very specific about what these compounds are and why they smell the way they do. At the molecular level, these are simple structures. Hydrogen sulfide is just two hydrogen atoms bonded to one sulfur atom. Methylmercaptan is a methyl group bonded to sulfur and hydrogen, but their simplicity makes them volatile. They evaporate easily at room temperature. They diffuse rapidly through air and water. They bind to olfactory receptors in your nose with extreme efficiency. Evolution tuned human noses to detect sulfur compounds at incredibly low concentrations, because throughout history, sulfur smells have indicated danger. Rotting meat, decaying bodies, spoiled food, toxic gases from volcanic vents. Our ancestors who could smell sulfur compounds at parts per billion concentrations and avoided them survived longer. Those who couldn't detect these warning signs died younger. So your disgust at morning breath isn't cultural, it's evolutionary. Hydrogen sulfide is the same gas that gives rotten eggs their signature stench. It's colorless, it's flammable. At high concentrations above 400 parts per million, according to the Centers for Disease Control and Prevention, it's dangerous enough to cause loss of consciousness within 30 minutes to an hour. Your mouth doesn't produce anywhere near that level, but even at low concentrations measured in parts per billion, the human nose can detect it. Research published in Oral Microbiology and Immunology in 1990 by Persson and colleagues from Sweden tested 75 oral bacterial species and 7 unnamed bacterial taxa. They found that the majority produced significant amounts of hydrogen sulfide from L-cysteine. The most active producers were found in genera including Peptostreptococcus, Eubacterium, Selenomonas, Centipeda, Bacteroides, and Fusobacterium. When they incubated these bacteria in human serum for 7 days, the most potent hydrogen sulfide producers were Treponema denticola and several black pigmented species including Porphyromonas gingivalis and Porphyromonas endodontalis. Methylmercaptan is worse. It's also called methanethiol. The chemical safety data from the National Institute for Occupational Safety and Health states that some investigators found toxicity levels similar to hydrogen sulfide, while others found it somewhat less toxic. Either way, it's nasty. The smell is described in the literature as rotten cabbage, putrid, and garlic-like. The natural gas industry adds methyl mercaptan to natural gas specifically because its smell is so penetrating and recognizable that people notice even tiny leaks. A 2014 incident at a DuPont facility in La Porte, Texas, released 11,000 kilograms of methyl mercaptan. It killed 4 people. Methyl mercaptan is formed when oral bacteria break down L-methionine. Research in the same 1990 paper by Persson found that fewer bacterial species produce methyl mercaptan compared to hydrogen sulfide. The producers included some members of Fusobacterium bacteroides, Porphyromonas, and Eubacterium genera. A separate 1991 study published in the Journal of Periodontology using gas chromatography found methyl mercaptan in about 20% of the 79 periodontal pockets they sampled with a maximum concentration of 0.16 millimoles per liter. Hydrogen sulfide was found in 61 pockets out of 79 with maximum concentrations reaching 1.9 millimoles per liter. Dimethyl sulfide rounds out the trio. It smells like cooked cabbage asparagus, or in wine chemistry literature where it's also studied as a fault compound like cooked corn. At lower concentrations, some describe it as having a truffle-like character, but in your morning breath, it's just adding to the overall foulness. Here's what makes this chemistry particularly disturbing. These volatile sulfur compounds aren't just smelly, they're biologically active. Research published in Oral Diseases in 1984 studied how hydrogen sulfide and methyl mercaptan affect oral tissue permeability using porcine non-keratinized sublingual mucosa. Exposure to hydrogen sulfide increased tissue permeability by up to 75%. Methyl mercaptan was worse at 103% increase. The researchers concluded that these compounds react with tissue components and alter the integrity of the tissue barrier. Radioactive tracers showed that hydrogen sulfide could penetrate intact layers of non-keratinized epithelium basal membrane and connective tissue. This means the bacteria aren't just making smells, they're producing molecules that can damage your oral tissues. The peak production happens during sleep because of the perfect storm of conditions. Saliva flow drops to near zero, eliminating the washing and oxygen delivery system. Your mouth temperature stays steady around 37 degrees Celsius, ideal for bacterial metabolism. You're not eating or drinking, so there's no physical disturbance of the bacterial biofilms coating your tongue and teeth. The bacteria have hours, 6 to 8 hours depending on your sleep duration, to multiply and metabolize. Think about what happens in those hours. Bacteria reproduce through binary fission. One cell splits into two, two become four 4 become 8. Generation times for oral bacteria vary. Fast-growing species like some streptococci can divide every 20 to 30 minutes under ideal conditions. That means one bacterial cell at bedtime could theoretically produce over 4,000 descendant cells by morning if nothing stopped it. In reality, growth isn't that explosive because nutrients become limiting and waste products accumulate, but the principle holds. Bacterial populations during sleep don't just maintain, they expand. The biofilm itself changes structure overnight. During the day, saliva flow and mechanical action from eating, talking, and swallowing keep biofilms thin and disrupted. At night, undisturbed, the biofilm thickens. Bacteria secrete extracellular polymeric substances, sticky matrices of polysaccharides, proteins, and nucleic acids. These matrices protect bacteria from environmental stress. They create microenvironments within the biofilm. The outer layers exposed to whatever oxygen remains stay aerobic. The inner layers pressed against your tongue or tooth surface become completely anaerobic. Oxygen can't penetrate the thick biofilm. The anaerobic species that produce volatile sulfur compounds thrive in these oxygen-depleted zones. They're shielded from saliva's antimicrobial effects by the biofilm matrix. They're surrounded by other bacteria in complex communities where different species cooperate. Some species process complex proteins into simpler peptides. Other species break peptides into amino acids. Still others metabolize those amino acids into volatile sulfur compounds. It's an assembly line of decomposition, and the end products, those sulfur gases, accumulate in the spaces between bacterial cells within the biofilm and diffuse out into your saliva and breath. Understanding all this makes you realize morning breath isn't a hygiene failure. It's human biology, but that doesn't mean you're helpless. The science that reveals the mechanisms also points to interventions. Subscribe if you're finding this detailed biochemistry valuable. Comment your location. And this is your second reminder that everything here comes from actual published research, including studies from BMC Microbiology in 2020, examining whole genome sequencing of the healthy oral microbiome, finding extensive antibiotic resistance genes, even in healthy mouths, and documentation from the British Dental Journal in 2016, reviewing how humans and oral microbes have co-evolved for millions of years in mostly harmonious symbiosis until modern diets and lifestyles disrupted that balance. Now continuing. A December 2021 study in PLOS ONE by Japanese researchers examined exactly how sleep changes the oral microbiome. They recruited 10 healthy subjects and professionally cleaned their teeth and oral mucosa 7 days before sample collection, then again 24 hours before. They collected samples from 6 locations: buccal mucosa, hard palate, tongue dorsum, gingival mucosa, tooth surface, saliva. They did this twice per subject, once right after waking and once right before sleep. The 16S ribosomal RNA sequencing revealed that biofilm-forming bacteria at oral sites belong to 5 major phyla: Actinobacteria, Bacteroidetes, Firmicutes, Fusobacteria, Proteobacteria. Firmicutes dominated the soft tissues. About 50 to 60% of bacteria on buccal mucosa hard palate and gingival mucosa were firmicutes. But on the tongue and in saliva, firmicutes made up only 27 to 32%. More importantly, the relative abundance of specific genera changed between night and morning. Prevotella on the buccal mucosa, hard palate, tongue, dorsum, and in saliva was significantly higher in the morning than at night. Statistical analysis using the Wilcoxon rank sum test gave p-values of 0.013 for buccal mucosa, 0.028 for hard palate, 0.013 for tongue dorsum, and 0.011 for saliva. Corynebacterium was more abundant in the morning on buccal mucosa, hard palate, gingival mucosa, and supragingival dental biofilm, with p-values ranging from 0.013 to 0.031. These shifts represent the bacterial community responding to the overnight environmental changes. Low oxygen, no saliva flow, no mechanical disturbance. The coating on your tongue deserves special attention. A January 2025 paper in the Saudi Dental Journal called it a biofilm comprising desquamated epithelial cells, food debris, and microorganisms. Studies estimate the thickness can range from 0.1 to 0.2 millimeters. At that thickness, the coating can deplete oxygen from the environment beneath it, creating anaerobic conditions. Our research shows a direct correlation between the amount of coating on a person's tongue and the total number of anaerobic bacteria present. More coating equals more anaerobic bacteria equals more volatile sulfur compound production. Different methods exist for quantifying tongue coating. The Winkle Tongue Coating Index developed in 2003 scores the tongue surface. Photographic analysis provides objectivity. Wet and dry weight analysis measures the actual mass of material scraped from the tongue. But the newest technique, published in December 2024, uses bacterial biofluorescence. The technology detects endogenous porphyrins. These are metabolic products of anaerobic bacteria. When you shine blue light at 405 nanometers on the tongue, dorsum, the anaerobic bacterial biofilms fluoresce red. The intensity and coverage correlate with hydrogen sulfide and methyl mercaptan levels. 81 elderly subjects provided 162 images. The researchers developed the tongue biofilm fluorescence index, scored on a 0 to 2 scale for both intensity and coverage. It showed better inter-examiner agreement than older visual methods. Now here's where it gets even more specific. The exact bacterial species matter. A 2016 study in Scientific Reports examined the salivary and tongue-coating microbiomes in children with halitosis versus healthy children using 16S ribosomal RNA pyrosequencing and metagenomic sequencing. They found that subjects with halitosis had greater bacterial richness on the tongue coating than healthy subjects. Two species stood out: Leptotrichia wade and Peptostreptococcus. Stomatis had higher relative abundance and prevalence in tongue-coating samples from children with halitosis. Prevotella shahi had higher relative abundance in saliva samples from children with halitosis. These aren't random bacteria. They're specific halitosis-associated pathogens that thrive in the conditions created during sleep. The production rate of these volatile sulfur compounds has been measured. Studies using gas chromatography, coupled with sulfur chemiluminescent detection, can quantify hydrogen sulfide methylmercaptan and dimethyl sulfide in parts per billion, the detection threshold. The level at which humans can smell these compounds varies by study. For methylmercaptan, the sensory threshold reported in wine chemistry literature ranges from 0.02 to 2 micrograms per liter. That's parts per billion. The human nose is exquisitely sensitive to these sulfur compounds. Evolution probably tuned this sensitivity because sulfur compounds often indicate decay, spoilage, and danger. Breathing through your mouth makes everything worse. Mouth breathing during sleep is associated with sleep-disordered breathing and blocked nasal passages. When you breathe through your mouth all night, the airflow dries your mouth even more. Saliva that might have stayed as a thin protective film evaporates. Bacterial counts climb higher. A 2002 study published in the journal Sleep found that 23% of Canadians self-reported dry mouth during sleep associated with awakening and water intake. The biological significance of decreased saliva during sleep remains unclear according to the authors. But the oral cavity clearly struggles to compensate for this period of relative dryness. Certain medications make the problem worse. A wide range of drugs produce dry mouth as a side effect. Antihistamines, decongestants, diuretics, antidepressants, blood pressure medications. When you combine reduced saliva during sleep with drug-induced hyposalivation, the bacterial proliferation accelerates. People on multiple medications face compounded risk. The literature documents that being on more than one medication increases dry mouth risk significantly. Smoking and tobacco reduce saliva production. The toxins in cigarettes and chewing tobacco affect salivary flow rate, making users produce less saliva than non-smokers. This creates baseline dry mouth that worsens dramatically during sleep. Radiation therapy for head and neck cancers can damage the nerves controlling salivation. Chemotherapy drugs also reduce saliva production. Diabetes affects saliva composition and flow. Poorly controlled blood sugar means salivary glands produce less saliva. Stress and anxiety decrease saliva production through sympathetic nervous system activation. Age plays a role. Elderly individuals often have reduced salivary flow for multiple reasons. Age-related changes in gland function, more medications, more systemic diseases. A 2025 study looking at tongue coating in hemodialysis patients found that thick tongue coatings correlate with oral microbial overgrowth, including anaerobes and possibly yeast. These microbes and their metabolites get continuously swallowed, potentially influencing gut microbiota composition downstream. The oral and gut microbiomes can mirror each other. Changes in oral microbiota accompany changes in gut microbiota in various diseases. The question becomes, what actually happens to these volatile sulfur compounds when you wake up and start your morning routine? Drinking water helps. It rehydrates tissues and stimulates some saliva production. Eating breakfast triggers massive saliva production. Chewing increases saliva flow to 3.15 to 4.94 milliliters per minute, according to research measuring flow during consumption of several different foods. Taste stimulation is even more effective. Infusing 5% citric acid into the mouth produces flow rates of 7.07 milliliters per minute. This flood of fresh saliva washes away bacterial waste products. The increased oxygen levels suppress anaerobic bacterial metabolism. The antimicrobial proteins in saliva start killing bacteria. Mechanical action from chewing and tongue movement dislodges biofilms. Your body essentially resets the oral environment within minutes of waking. The bacterial populations don't disappear, but they're pushed back to manageable levels. The volatile sulfur compounds get diluted and swallowed or exhaled away. Brushing your teeth removes bacterial biofilms from teeth surfaces. The physical scrubbing action with bristles disrupts the extracellular matrix that holds biofilms together. Toothpaste adds chemical antimicrobials. Fluoride not only strengthens enamel but also inhibits bacterial enzymes. Sodium lauryl sulfate in most toothpastes is a detergent that helps remove debris and has some antimicrobial properties. Triclosan, in some formulations, directly kills bacteria, though its use has become controversial. Flossing removes food particles and bacteria from between teeth, where toothbrush bristles can't reach. Approximately 38% of tooth surfaces are interdental. You can't clean them without floss or interdental brushes. Tongue scraping or tongue brushing removes the coating layer where anaerobic bacteria concentrate. Studies show daily scraping or brushing reduces the substrate for putrefaction rather than reducing bacterial load directly. The bacteria repopulate quickly, but removing their food source and metabolic waste products improves breath immediately. One study found that tongue cleaning reduced volatile sulfur compound levels by 75% immediately after cleaning. The effect persists for several hours before bacterial metabolism ramps up again. Using mouthwash kills bacteria chemically. Alcohol-free formulations with antimicrobial agents like chlorhexidine or cetylpyridinium chloride reduce bacterial counts. Chlorhexidine at 0.12% concentration can reduce oral bacteria by over 90% within minutes. However, long-term chlorhexidine use has side effects: tooth and tongue discoloration, bad taste, decreased taste sensation. Zinc chloride at 0.22% concentration can neutralize methyl mercaptan by forming zinc salts. The zinc ions bind to the sulfur atoms in methyl mercaptan, creating non-volatile zinc mercaptide complexes that don't smell. The metabolic pathways bacteria use to produce these compounds have been mapped. Several 2025 papers examined tongue-coating metabolic profiles using liquid chromatography mass spectrometry. They identified 2,214 metabolites in tongue-coating samples. Comparing halitosis patients to healthy controls revealed that several metabolic pathways were significantly enriched in halitosis subjects: arginine biosynthesis, arginine and proline metabolism, histidine metabolism, lysine degradation. These pathways involve breaking down amino acids, some of which contain sulfur. The bacteria use enzymes to cleave the amino acids, releasing the volatile sulfur compounds as waste products. Researchers found that paramethasone acetate and indole 3-acetic acid were upregulated in halitosis patients. These are metabolic markers, chemical signatures of the bacterial community's activity. The bacteria aren't just making smells, they're running complex biochemical factories, processing proteins into peptides, peptides into amino acids into smaller molecules, including the volatile sulfur compounds. Gender differences exist. A 2001 study mentioned in the Dutch journal International Journal of Oral Science noted that women manifest higher volatile sulfur compound levels than men in the morning. The mechanism isn't fully understood. Hormonal influences on bacterial communities, differences in salivary composition, differences in oral microbiome structure. The phenomena needs further investigation, but it's documented. Diet affects morning breath. Foods containing sulfur compounds like garlic and onions don't just leave residues in your mouth. They're absorbed into your bloodstream and eventually exhaled through your lungs and excreted through saliva. This creates blood-borne halitosis that combines with the bacterial production of volatile sulfur compounds. Protein-rich diets provide more substrate for bacterial metabolism. Carbohydrates feed different bacterial species, but these tend to produce acids rather than volatile sulfur compounds, leading to different issues like dental caries. Periodontal disease amplifies morning breath significantly. When you have gum disease, the pockets between teeth and gums become deeper. Normal gingival crevices are less than 3 millimeters deep. Periodontal pockets exceed 3 millimeters. These pockets harbor massive bacterial communities protected from saliva flow and mechanical cleaning. The bacteria in periodontal pockets include many of the same anaerobic species that colonize the tongue: Porphyromonas gingivalis, Treponema denticola, Tannerella forsythia, Fusobacterium nucleatum. Studies show that methylmercaptan is the greatest contributing volatile sulfur compound in halitosis, caused by periodontal disease and gingivitis. The level of volatile sulfur compounds on breath correlates positively with the depth of periodontal pockets, the number of pockets, and whether the pockets bleed when examined. The treatment of periodontal disease through scaling and root planing can improve mouth odor considerably. Removing subgingival calculus, tartar, hard plaque, and friable disease tissue reduces the bacterial reservoir. Irrigation with antibiotic mouth rinses suppresses the anaerobic bacteria temporarily, but without ongoing maintenance, the bacteria recolonize within days to weeks. Some people have pseudohalitosis. They believe they have bad breath, even though objective measures and other people detect nothing abnormal. This is estimated to make up between 5 and 72% of halitosis complaints, depending on the study. It's a psychological condition sometimes called delusional halitosis or monosymptomatic hypochondriasis. These individuals need reassurance and sometimes psychiatric support rather than dental treatment. For the rest of us, the majority, morning breath is physiological. It's normal. Every healthy person experiences it to some degree because every healthy person's saliva production drops during sleep, and every healthy person has anaerobic bacteria in their mouth. The question isn't whether you'll have morning breath. The question is how severe it is and what you can do about it. Interventions focus on 3 strategies: reduce bacterial load, reduce substrate availability, disrupt the anaerobic environment. Reducing bacterial load means mechanical cleaning, brushing twice daily for 2 full minutes. Most people think they brush for 2 minutes, but studies show the average is 35 to 45 seconds. Flossing daily, using antimicrobial mouthwash, cleaning the tongue either with a scraper or with a soft brush and oxygenating toothpaste. Professional dental cleanings every 6 months or more frequently if you have periodontal disease. Reducing substrate availability means removing food particles, not eating or drinking anything except water before bed. If you wake during the night, drinking water moistens the mouth and dilutes bacterial waste products. Staying hydrated throughout the day ensures better saliva production. Disrupting the anaerobic environment means increasing oxygen. Saliva naturally contains dissolved oxygen, which directly kills some anaerobic bacteria. Chewing sugar-free gum stimulates saliva flow. The flow rate with gum chewing peaks at about 6 milliliters per minute in the first minute, then decreases to a plateau around 1 milliliter per minute, well above the normal unstimulated flow rate of 0.3 to 0.4 milliliters per minute. This elevated flow can be maintained for 2 hours or more. Using a humidifier at night keeps bedroom air from becoming too dry, which helps prevent mouth-breathing related dryness. Treating underlying conditions matters. If you have sleep apnea, the constant mouth opening dries your mouth severely. CPAP therapy helps, but many CPAP users still experience dry mouth. Adding a humidifier to the CPAP device reduces dryness. If you have chronic nasal congestion treating, it reduces mouth breathing. Allergies, deviated septum, chronic rhinosinusitis, whatever the cause, fixing nasal breathing improves mouth moisture. For severe dry mouth, prescription medications exist. Pilocarpine and cevimeline are FDA-approved systemic sialogogues. They stimulate muscarinic receptors in salivary glands, increasing saliva production. Pilocarpine is typically prescribed at 5 milligrams 3 times daily for at least 3 months. Cevimeline has stronger affinity for M3 muscarinic receptors. Both provide similar benefits, but only work if you have functional glandular tissue remaining. If radiation therapy destroyed your salivary glands, these drugs can't help. In those cases, artificial saliva products and frequent water sipping are the main options. The prevalence of halitosis worldwide ranges from 15 to 60% of the population, depending on the study and definition used. A German study in the city of Bern found that self-reported halitosis was common, but clinical measurements often showed lower prevalence. Cultural factors affect willingness to report the condition. Social stigma makes discussion difficult. The economic impact is substantial. The market for breath freshening products runs into billions of dollars annually. Mints, gum, mouthwash, tongue scrapers, specialty toothpastes, dental visits specifically for halitosis complaints, time lost at work due to self-consciousness, social isolation, psychological distress. A 2012 review in International Journal of Oral Science by German researchers Kurt Bollen and Thomas Beichler concluded that halitosis affects some 25% of the world population and has a large social and economic impact. They noted that halitosis is a poorly researched disease mainly due to social taboos. The gold standard for measuring halitosis is organoleptic evaluation. A trained examiner smells the patient's breath and scores it on a 6-point intensity scale. This seems primitive, but it's more reliable than many instrumental methods because human olfaction is extraordinarily sensitive to sulfur compounds. Portable sulfide monitors called halometers measure volatile sulfur compounds in parts per billion. Gas chromatography is more sophisticated, measuring hydrogen sulfide, methyl mercaptan, and dimethyl sulfide separately. The oral chroma device uses gas chromatography, specifically designed for chairside use in dental offices. The connection to systemic diseases is real, but often overstated. 80 to 90% of halitosis cases are intraoral. They come from the mouth. The remaining 10 to 20% are extraoral, respiratory diseases, sinusitis, tonsillitis, lung infections, gastrointestinal disorders, gastroesophageal reflux disease, Helicobacter pylori infection, metabolic disorders, uncontrolled diabetes producing ketones that smell like rotten apples, liver failure, kidney disease producing uremic breath, certain medications producing characteristic odors, but for morning breath specifically. For that universal experience of waking up with a foul taste and smell, the cause is overwhelmingly local. It's the bacteria in your mouth feasting on available proteins during the 8 hours when your saliva stopped flowing. Some bacterial species deserve special mention for their role. Solobacterium morae, a gram-positive anaerobic rod. It's been isolated from tongue coatings of people with severe halitosis. Adipobium parvulum, another gram-positive anaerobe associated with halitosis. Eubacterium species, particularly Eubacterium sulci. These bacteria are adapted to the oral environment. They've evolved mechanisms to adhere to oral surfaces, to resist the limited antimicrobial effects of saliva, to survive periods of nutrient scarcity, to rapidly exploit the feast conditions that occur during sleep. The bacterial load increases dramatically overnight. Estimates vary, but some studies suggest bacterial counts in saliva can increase by 10-fold or more during sleep. The tongue coating weight can increase measurably. When researchers scrape tongues and weigh the material before bed versus after waking, the difference is significant. Evolutionary perspective matters. Why did humans evolve to have this problem? Several factors. The oral microbiome serves beneficial functions. Many oral bacteria are commensals, or even beneficial. They occupy niches that might otherwise be colonized by pathogens. They metabolize nutrients. They interact with the immune system in ways that train it. The price we pay is that some of these bacteria produce smelly compounds as metabolic byproducts. The reduction in saliva flow during sleep evolved for water conservation and uninterrupted sleep. Early humans, sleeping in environments where water was scarce and where waking repeatedly would be dangerous, the benefits of water conservation and sleep consolidation outweighed the cost of morning breath. Besides, morning breath isn't dangerous, it's just unpleasant. Modern humans have solutions our ancestors lacked: running water, toothbrushes, toothpaste with fluoride and antimicrobials, dental floss, mouthwash, professional dental care. We can manage morning breath easily. Brush your teeth before bed. Brush your tongue. Floss. Use mouthwash. Don't eat after brushing. Stay hydrated. Treat any periodontal disease. Address nasal congestion. In the morning, brush and floss again before eating. Drink water. Your saliva production will resume. Your breath will normalize. The chemistry paper published in the Journal of Agricultural and Food Chemistry in 2017 by Kreitman and colleagues explored the fate of hydrogen sulfide and methanethiol during wine storage. Why does this matter? Because the same volatile sulfur compounds in your mouth are faults in wine. The wine industry spends enormous resources understanding and controlling these compounds. They've mapped the equilibria. Hydrogen sulfide and methyl mercaptan exist in free volatile forms. They also bind to metal ions like copper-forming complexes. They oxidize to form disulfides and polysulfides. These forms interconvert through reversible redox reactions. Wine redox potential determines which forms dominate. In oxidizing conditions, the mercaptans convert to non-volatile disulfides. In reducing anoxic conditions, the disulfides reduce back to smelly mercaptans. Your mouth during sleep is a reducing environment. Low oxygen. Anaerobic bacteria consuming whatever oxygen remains. Perfect for maintaining the reduced, smelly forms of these sulfur compounds. The relationship between tongue coating and systemic health extends beyond breath. A 2025 study in Frontiers in Cellular and Infection Microbiology examined hemodialysis patients. Those with thick tongue coatings showed gut microbiome dysregulation. The oral bacteria and their metabolites get swallowed continuously. They influence the gut microbiota. In diseases including kidney disease, inflammatory bowel disease, cardiovascular disease, the oral gut axis appears important. Tongue coating isn't just about breath. It's a window into overall microbial ecology. Cancer connections exist. Some studies link certain oral bacteria to increased risk of oral cancer, esophageal cancer, pancreatic cancer, colorectal cancer. The mechanisms aren't fully understood, but chronic inflammation and bacterial metabolites that damage DNA are suspects. This doesn't mean morning breath causes cancer. It means the bacterial communities in our mouths are important for reasons beyond breath odor. Aspiration pneumonia in elderly or disabled individuals can result from oral bacteria entering the lungs. When swallowing reflexes are impaired, oral bacteria mixed with saliva get aspirated into the respiratory tract. If bacterial load in the mouth is high and includes pathogenic species, aspiration can cause severe pneumonia. This is why oral hygiene in hospital and nursing home settings is critical. The take-home message about morning breath is this: it's a normal physiological phenomenon resulting from reduced saliva flow during sleep, creating conditions where anaerobic bacteria metabolize proteins into volatile sulfur compounds, including hydrogen, sulfide, methyl mercaptan, and dimethyl sulfide. These compounds smell terrible. They can damage tissues at high concentrations. They indicate bacterial metabolism. You can reduce morning breath severity through good oral hygiene, mechanical removal of bacteria and their substrate, maintaining hydration, treating periodontal disease, addressing factors that reduce saliva, like medications, mouth breathing, and smoking, using antimicrobial mouthwash appropriately, cleaning your tongue daily, but you can't eliminate morning breath completely without eliminating your oral microbiome, and you don't want to do that. Your oral microbiome, despite producing smelly compounds overnight, provides benefits. It occupies ecological niches. It interacts with your immune system. It's part of being human. The weirdest part of morning breath isn't that it happens. It's that the same molecules responsible, hydrogen sulfide and methyl mercaptan, are toxic industrial chemicals at high concentrations, fatal at parts per million. Yet your body produces them every single night in parts per billion from normal bacterial metabolism. Your nose detects them at incredibly low levels because evolution tuned your olfactory system to recognize decay, and every morning you wash them away and start fresh, until night falls and the cycle repeats. In 2016, researchers published a detailed analysis of exactly which bacterial species correlate with halitosis in children using cutting-edge sequencing. In 2021, Japanese researchers tracked how bacterial communities shift from night to morning across 6 oral sites in 10 subjects. In 2024, Korean researchers developed fluorescence imaging to visualize anaerobic bacteria on tongues. In 2025, scientists published updated global averages for healthy salivary flow rates from over 3,000 adults. The picture keeps getting sharper, the mechanisms more precise, the species identified down to strain level. This isn't folklore. This isn't guesswork. This is documented, repeatable, quantified science. Gas chromatography, measuring compounds in parts per billion. DNA sequencing, identifying bacterial species to over 99% accuracy. Statistical analysis with p-values and confidence intervals. Microscopy, showing biofilm structure. Chemical analysis, mapping metabolic pathways. The knowledge exists. The interventions work. The chemistry is understood. And yet most people wake up, taste something terrible, brush their teeth, and never think about the fact that overnight their mouth became a bacterial reactor, producing industrial chemicals from dead cells and food residue, while their saliva production dropped to near zero, creating perfect anaerobic conditions for gram-negative bacteria to metabolize cysteine and methionine into hydrogen, sulfide, and methyl mercaptan through enzymatic pathways that have been mapped at the molecular level. That's morning breath. Not a mystery. Not a curse. Not poor hygiene. It's human biochemistry, bacterial ecology, evolutionary compromise, and the price we pay for sleeping through the night without drowning in our own saliva. A mass grave of epithelial cells, a feast for bacteria, a chemical factory running on autopilot, and every morning you clean it up and start again. The cycle never stops. Not as long as you're breathing. Not as long as you're human. The bacteria were here before you. They'll be here after you. And every night while you sleep, they'll keep making those molecules. Keep filling your mouth with that smell. Keep reminding you that you're not alone in your body. You're an ecosystem, a walking, talking bacterial amusement park, and morning breath. That's just the overnight shift clocking out. But here's what makes this story even stranger. Recent research is revealing that your oral microbiome doesn't just affect your breath, it affects your entire body. When you swallow, which happens roughly 500 to 1,000 times per day, you're sending oral bacteria and their metabolites into your digestive system. Studies tracking bacterial DNA have found oral species in stomach biopsies, intestinal samples, and even in feces. The oral gut axis, as researchers call it, appears to influence conditions ranging from inflammatory bowel disease to metabolic disorders. A 2024 study examining tongue coating thickness in hemodialysis patients found that those with thick coatings had significantly different gut microbiomes compared to those with thin coatings. The oral bacteria they swallow daily were reshaping their intestinal microbial communities. Other research has linked specific oral bacteria to cardiovascular disease. When Porphyromonas gingivalis, the periodontal pathogen, enters the bloodstream through inflamed gums, it can travel to arterial plaques. Researchers have found bacterial DNA from oral species in atherosclerotic lesions. The chronic low-grade inflammation driven by periodontal bacteria may contribute to heart disease risk. Cancer connections keep emerging. Studies have associated certain oral bacterial profiles with increased risk of pancreatic cancer, esophageal cancer, and colorectal cancer. The mechanisms aren't fully mapped, but researchers suspect that bacterial metabolites that damage DNA and chronic inflammation create conditions favoring malignancy. Fusobacterium nucleatum, one of the bacteria that produces hydrogen sulfide in your mouth, has been found enriched in colorectal tumor tissue. Its exact role in cancer development remains under investigation, but the correlation is striking. In elderly and disabled individuals, oral bacteria can cause life-threatening aspiration pneumonia. When swallowing reflexes falter, oral bacteria mixed with saliva get inhaled into the lungs. If the bacterial load is high and includes pathogenic species like Streptococcus pneumonia or Staphylococcus aureus, which can colonize the mouth, the result is severe infection. This is why oral hygiene in hospital and nursing home settings has become recognized as critical preventive care. Daily tooth and tongue cleaning in ventilated patients reduces pneumonia rates by roughly 40%, according to meta-analyses of clinical trials. The Human Oral Microbiome Project, which ran from 2007 to 2017, revealed that an enormous number of bacteria inhabit the human body, forming indigenous microbiomes in each habitat. The oral microbiome turned out to be far more complex than anyone expected. Different sites in your mouth harbor distinctive communities: your tooth surfaces, your tongue, your cheeks, your hard palate, your gums. Each supports different bacterial assemblages. Some species are generalists found everywhere. Others are specialists restricted to specific niches. The project cataloged the species, sequenced their genomes, mapped their metabolic capabilities, created databases that researchers worldwide now use. And what they found is that healthy mouths have bacterial diversity. Disease states, whether caries or periodontitis or halitosis, involve loss of diversity and overgrowth of specific pathogenic species. The shift from health to disease isn't about eliminating bacteria, it's about maintaining balance. Modern lifestyles disrupt that balance. Sugar consumption provides easily fermented substrates that acid-producing bacteria like Streptococcus mutans exploit. The bacteria adapted. Streptococcus mutans developed more efficient carbohydrate metabolism, defenses against oxidative stress. Resistance to acidic environments, it creates itself. These adaptations allowed it to outcompete other species and dominate. Frequent snacking maintains high sugar availability, preventing the mouth from recovering its neutral pH between meals. Antibiotics kill susceptible bacteria, but spare resistant strains, creating selection pressure for resistance genes. Studies using whole genome sequencing on oral bacteria from healthy individuals found extensive antibiotic resistance genes, even in people who hadn't taken antibiotics recently. The resistance genes spread through bacterial communities via horizontal gene transfer. One bacterium passes resistance genes to neighbors through plasmids or phages. The oral cavity becomes a reservoir of resistance genes that can potentially spread to pathogenic bacteria elsewhere in the body. Smoking reduces saliva production and damages oral tissues, creating more niches for pathogenic bacteria. Stress alters saliva composition and reduces flow through sympathetic nervous system activation. Poor sleep disrupts circadian rhythms that influence bacterial gene expression. Research published in 2021 found that oral bacteria exhibit circadian patterns in their metabolic activity linked to host behaviors like eating and sleeping. Disrupted sleep schedules disrupt these patterns, potentially favoring dysbiosis. The oral microbiome is dynamic. It responds to diet, to hygiene practices, to systemic health status, to medications, to age. Infants acquire their initial oral bacteria from their mothers during birth and breastfeeding. Children's oral microbiomes diversify as they age and their diet expands. Adult microbiomes stabilize but remain responsive to environmental changes. Elderly individuals often show reduced diversity, possibly related to medications, dry mouth, and systemic diseases. Understanding morning breath in this broader context reveals it as one small symptom of a vast and complex microbial ecosystem. The smell you notice when you wake is just the visible tip of biochemical processes involving hundreds of bacterial species, dozens of metabolic pathways, and millions of individual cells. Those processes don't stop with volatile sulfur compounds. Oral bacteria produce acids that demineralize enamel, proteases that degrade gum tissue, lipopolysaccharides that trigger inflammation, adhesins that help them stick to surfaces, bacteriocins that kill competing species. They form cooperative communities where different species trade nutrients. They communicate through chemical signals. They adapt to antibiotics and antimicrobials. They persist through daily disruptions. They've been evolving alongside humans and our ancestors for millions of years. And they'll continue evolving. Every time you use mouthwash, you're applying selection pressure. Every time you eat sugar, you're feeding certain species more than others. Every time you skip brushing, you're allowing biofilms to mature undisturbed. You're not just a passive host. You're actively shaping your oral microbiome through your choices. The research continues accelerating. New technologies allow scientists to study not just which bacteria are present, but what they're doing. Metatranscriptomics sequences bacterial RNA revealing active gene expression. Metaproteomics identifies proteins bacteria are producing. Metabolomics measures the small molecules bacteria generate. These approaches paint increasingly detailed pictures of oral microbial metabolism. Researchers can now see that during sleep, certain bacterial genes get upregulated, while others get downregulated. They can measure exactly which proteins bacteria produce in response to changing oxygen levels. They can track how bacterial metabolites interact with human cells, triggering immune responses or damaging tissues. The complexity is staggering. Your mouth contains an entire miniature ecosystem, multiple habitats, hundreds of species, trillions of individual organisms, complex food webs, predator-prey dynamics, competition, cooperation, evolution in real time, and morning breath is what it smells like.